Today we are working for the first time on summer 2018, paper 21. So the idea here is to work through the questions as we get them, mixed topic, mixed technique, and see what happens when we work through a full paper. So it's a paper 2, it's a short answer questions. You're answering all the questions, you're answering them in the space provided. Starting out with question 1. Figure 1.1 is a transmission electron micrograph of a cell from the root of tail cress, Arabidopsis thaliana. We have pretty obvious cell wall, which is lovely. Might help if I plug my pen in. Okay, right, we are in business. Pretty obvious cell wall running down the outside here. Okay, then we can see various things going on inside the cell, and we have a few things labeled. So A part 1 asks us, the structures labeled A and B on figure 1.1 are sections of two mitochondria. Suggest why A and B are different shapes. So you see A is a circle, whereas B is a sort of sausage-shaped oval. And what we've got to do is we've got to think about structure, the 3D structure of a mitochondrion. So we always draw our mitochondrion like this. But remember, it is actually rather like a sausage shape. So if we cut it in this direction, then we would have a cross-section of a circle. So when we're doing, when specimens are being prepared for electron micrographs, okay, so we're preparing the specimen to go into the electron microscope, then we're cutting very, very thin slices through the cell. So that is why when we're then cutting through the cell, the mitochondria are all over the place. And different mitochondria can be cut in different directions within one section. So here we can say the reason why A and B are different shapes is that they were cut. So the sections that were taken of that mitochondria are in different orientations. If you want to be a little bit more specific, A is a cross section. And B shows a longitudinal section. So one single organelle, but two different appearances because of the way it was cut. So it's always important to remember when you're studying cells, everything's happening in 3D. It's not just a simple line drawing like we see so often in books. So the structure labeled D on figure 1.1 is a mitochondrion about to divide. And there you can see there's a piece pinching off there. Explain the importance of the division of mitochondria for the cell shown in figure 1.1 and for cells in the root tips of tail cress. So this cell here, remember, is just a root cell, okay? Not necessarily a root tip. So here we're talking about root plus root tip. So why would we need division of mitochondria in a general cell and in a root tip in particular as well? So what is the purpose of a mitochondria? Okay. The purpose of a mitochondria is aerobic respiration. So with production of ATP, which is used by the cell. So the more mitochondria we have, the more energy we have. So as the cell in the root grows, we're going to need more mitochondria to support the bigger cell. In the root tip, remember, this is a region of cell division. So here we would be looking at needing new mitochondria for new cells. Because when we divide into two daughter cells, both of those daughter cells are going to need mitochondria. In order to do that, we need to replicate the mitochondria. So we'll start with a sort of overall, what is the be-all and end-all for mitochondria? It's the production of energy. So mitochondria provide most of the energy. Remember, we do get some energy from anaerobic respiration, but the vast majority is coming from aerobic respiration in our mitochondria. Okay, so in the root, more mitochondria are needed as cells grow. And then specifically in the root tip, more mitochondria are needed for daughter cells. And if I was using a normal pen on a normal piece of paper, then I have no doubt this would fit within the required space. But just a little note here, that this sort of notes in the margin that I've done here is something that you can do in your exam as well. So you can prepare yourself for answering the question. The more marks, the more preparation, you get your things set in your head, and then you give a considered response rather than just regurgitating information that you hope might answer the question in some way. You want to rather aim for considered responses that have been thought about and planned. So moving on to section B. Within a cell, substances move between the nucleus and the cytoplasm. The area labeled C in figure 1.1 shows an area where this communication occurs. Before we even look at it, we know that we're talking about a nuclear pore. Make a large label drawing of area C to show where this communication occurs. So there is our C. If we zoom in, seeing as we're doing this digitally, and if we zoom right in to the point where the computer is not happy with it being so zoomed in, we can actually see the pore. So we can see that the membrane comes here, and then right in the middle there is a gap. So you can see it, that's your nuclear pore. So now if we zoom back out again to a more comfortable place to write, make a large label drawing of area C to show where this communication occurs. So we had the membrane coming down and back up, because remember it's a double membrane. It's a nuclear envelope. And then we had the other piece from the other side, cross section again. So this here would be the nuclear core, and this here is the nuclear envelope. And we want to label both of this because they've given, it said draw a large label drawing. So that is the instruction. So it's not like we're going to go added other detail. Okay, this is what's shown in C. But it's important that we want to label both the core and the envelope. So they didn't just say label the piece of communication. Okay? Label the drawing means that we want to then label everything. So sticking with the nucleus for question C, outline the functions of the nucleus in non-dividing cells, such as the cell in figure 1.1. Remember, this is just a standard root cell, no root tip, because our cell division is happening in like lots of, when we get to the root tip, we're just now back in the normal root. So what is the function of a nucleus? Biggest function, contain and protect the DNA. So we could say that the nucleus protects DNA from degradation. That's like the basic, but now we need to outline the functions. So we need to unpack this a little bit. So the nucleus contains chromosomes. Okay. And what is the function here? Well, the chromosomes have the coded instructions for polypeptide synthesis. Okay. However, we need to go a little bit further. We have transcription going on, okay, in the nucleus. That's a function that happens in the nucleus. So the sequence of bases in DNA is copied through transcription to create mRNA. So now we've gone actually from protection all the way into control of the cell's activities. So we also have an important area within the nucleus, and that's the nucleolus. So the nucleus contains the nucleolus. So let's go back again. So nucleus contains the nucleolus, which is the site of manufacture of ribosome subunits. So quite important functions. So those are all the different things that's happening in the nucleus, going from the overall arching of protection of the DNA down to, okay, so we're going to protect the DNA, but why is the DNA important? How does a cell then, how does the nucleus control the cell? And then never forgetting that the nucleolus is also an element of the nucleus with a very important function. So a total of 9 marks. So we're done with question 1, and we move then to question 2. And as soon as you see it, you know exactly which topic you're heading to. Biological molecules, here we come. So sucrose is a disaccharide. Figure 2.1 shows how sucrose is broken down in a reaction with hydrochloric acid. Well, we've done this because this forms part of our non-reducing sugars test. We break down with hydrochloric acid before we do a Benedict's. So we're happy so far. And there is our sucrose, and there are our 2 products. And in this case, they actually want us to name the products A and B of this reaction. So A is, of course, our good friend glucose. OH below, thus we know that this is now alpha glucose. And B, the other monosaccharide that combines to make sucrose, is fructose. So fructose plus alpha glucose makes sucrose. Name the type of bond that is broken in the reaction shown in figure 2.1. So what is this bond? A bond within a carbohydrate is a glycosidic bond. And remember that spelling for all of these sorts of things is critical. You must be comfortable with how to spell your technical terms. State the type of reaction shown in figure 2.1. We are adding water, so this is a hydrolysis reaction. When we remove water to build, it's condensation. When we add water to break, it's hydrolysis. Funnily enough, we're now moving on to Benedict's. So when Benedict's solution is added to a sucrose solution, a Benedict's plus sucrose, and put into a boiling water bath, no change in color is observed. State why no color change is observed. Well, sucrose is a non-reducing sugar, so it's not going to do anything, but it's 2 marks. So we need a little bit more detail. So we'll start with the basic and obvious. Sucrose is a non-reducing sugar. And remember here they said Benedict's plus sucrose. No hydrochloric acid anyway. So hydrochloric acid has not been used to break down the sucrose into reducing sugars. So if we want to get a little bit more technical, and as in where does that color come from, what do we actually mean by this idea of a reducing sugar? It's the fact that sucrose cannot reduce the blue copper 2 ions to red copper 1 ions. Now, maybe when you were initially studying the reducing sugars test and non-reducing sugars test, you didn't pick up on this. Okay, maybe you skipped over that bit and you thought, oh, that was a bit too much chemistry. I don't want to learn that. Thing is, there is nothing that you can't want to learn because you need to know all the information. But the other thing to bear in mind is as you do revision, as you work through past papers, if you come across things and you thought, oh, I could add that in now, go back to your notes, go back to your study summaries, and add in information from past paper memos. It's a wonderful source of learning, is just the information in those memos. So past papers are great for testing your knowledge. They're also great for adding in a few little bits and pieces that you maybe neglected on your first way through when you are learning the material to start with. So moving on to C. Sugar beet plants, Beta vulgaris, store sucrose in their roots. Describe and explain. So we need to describe and we need to give the reasons, the details, the technicalities. How sucrose is transported from the phloem in the leaves of sugar beet to storage tissues in the root. Right, so we've now got to step through a few things here. Okay, so we are in the phloem. Okay, well, where would we be in the phloem? To start with, we would be in the companion cell. So we've got to get from the companion cell to the sieve element. We've got to then build up pressure. Then we've got to get the pressure difference because of what's happening at the sink, the removal of that sucrose. And then we're going to get mass flow. So you see, just a few short notes can help you then structure your answer, and you don't sort of go off on a tangent within a longer answer. This is 4 marks. It's a relatively long answer for these papers. Okay, so companion cell to sieve element with some proper technical terms. So sucrose in companion cell diffuses into sieve tube element via plasmodesmata. Very important, diffusion and plasmodesmata. Okay, so what happens next? We affect the water potential. Water potential in sieve tube element is lowered. So water enters via Phloem is living tissue, so across the cell membrane via osmosis. Okay, so now we have a whole lot of water entering. We are increasing that hydrostatic pressure. Hydrostatic pressure in leaf increases. So then what happens? Well, we get mass flow, but we've got to prep as to why that mass flow, because we talked about this pressure difference. So we have low hydrostatic pressure in root as sucrose is removed and water leaves sieve tube element. So therefore, high pressure to low pressure, phloem sap moves by mass flow down the hydrostatic pressure gradient from the leaves to the roots. So we have covered now the description of how it moves in, it moves down, it moves out, and we've covered the explain. Why do these things happen? Why does it move like this? And we've covered describe and explain. We've filled our space, which is also a good indication that we have answered the question in full and that we are ready then to move to the next question. So this is a total of 10. So we finished with question 2. But also, if you just have a think back, we started this question in biological molecules. Okay, that was all about biological molecules, and then we went to plant transport. So as you're working through past papers, keep this in mind, that you're chopping and changing from one topic to another and different interlinks between the topics. And remember that the more linkages you find between topics, the easier they are to remember, because you're remembering them all in different directions rather than just one list of information. So you're building a body of knowledge rather than isolated pieces of information. Our next question, a graph. So researchers isolated a sucrase enzyme from the bacterium Bacillus subtilis. They immobilized the enzyme in alginate leaves. The researchers investigated the effects of temperature on the activity of the immobilized sucrase compared with the activity of the free enzyme, same enzyme, free in solution. The results are shown in figure 3.1. So we've got percentage of maximum enzyme activity against temperature in degrees C. We're looking at a range from 40 to 80 degrees C. With our X's, we've got our immobilized sucrase, and with our dots, we've got sucrase, which is free. Okay, and we can see that they both go up. We have 100% at the same point for both types of enzyme, and then they go down, but they go down with a gap here at the end. They are not the same. So they're not as similar at high temperatures as they are at the lower temperatures. Keeping that all in mind, moving on to the question. With reference to figure 3.1, compare the effects of temperature on the activity of immobilized sucrase with the activity of sucrase free in solution. And this is now 4 marks. So this is a question requiring proper attention to detail, and we have only a few minutes left here, so we shall continue this in our next past paper question lesson. We've also had a question come through referring back to this question. So let's just discuss this a little bit more. So the questions come through is, why does the sucrose move as well and not just the water? So if we use this space that they left us, okay, so sieve tube element, companion cell, then we hope the eraser is going to play nice games with us. Where's he gone? So now we've got plasmatic matter, we've got a sieve plates, so we can actually look at what we're talking about. Okay, so sieve plates on either end, plasmatic matter into the companion cell. Okay, and then we've got a sort of a thin layer of cytoplasm around the outside. So beautiful, beautiful drawing of thin layers. Of course, we've got cytoplasm as normal in here. And remember that cytoplasm is relatively jelly-like. Okay, so think of jelly and custard, like nice, thick, wobbly jelly. Okay? When we're now talking about these other things, we are talking about the sucrose. Okay, so let's get another color for sucrose. Let's put sucrose in green because it's product synthesis. So the sucrose comes in and it's a disaccharide. So there's our sucrose molecules. Okay. Then we're adding water. But we're adding a whole lot of water. So we're effectively making syrup. Okay, but now with a different viscosity to that of the cytoplasm. And this combination of sucrose plus water plus other things that happen in there is your phloem sac. Okay? So the sucrose is in solution in water, and therefore when the water moves, it's going to take everything with it. So it's the same way if you have a glass of Oros, if you pour the water out, the Oros goes too. The Oros doesn't stay in the glass and then the water pours off. Okay, so when we have things in solution, they're going to move together. So because the sucrose has gone in here and then the water has come in to effectively dilute that cell sap, so phloem sap, then it's all going to move as one liquid together. That makes sense. Brilliant. So we'll continue with this paper in another lesson.